And welcome everyone, and thank you for joining us today for our webinar titled High Throughput Investigation of EC Coupling in Isolated Cardiac Myocytes. This is Andy Henton from Inside Scientific, and I will be your host for today's event. Our session is sponsored by Ion Optics and will focus on new technology and methodology supporting fast data acquisition and analysis of cardiomyocyte function, which is proving to lead to better lab practice, data interpretation, and reveal novel scientific discoveries. To lead us through today's lecture, we are joined by Dr. Michael Helms. Over the past 20 years, Dr. Helms has been able to successfully combine academic research practice with an industrial career. After his PhD studying Titan mechanics in cardiac muscle with Dr. Henk Grenzier, he has kept various academic appointments, including positions at Boston University, the University of Oxford, and now the VU University Medical Center in Amsterdam in the Netherlands. Today, Dr. Helms also holds the position of Chief Scientific Officer at Ion Optics and is fully immersed in system development projects. His most recent project has been the design and refinement of a high-throughput system for calcium contractility measurements, which has been recently commercialized under his new company called Cytocipher. The design, operation, and resulting data offered by this new technology will be the focus of today's webinar.
Welcome, everybody. I saw that there were 280 people registered for this talk, which is a lot of people, so there must be a decent amount of interest. I see that you're all over from the world. A lot of people wanted to know about cardiac myocyte isolation in addition to other things. That's not what I'm going to talk about, although it's extremely important. But maybe we can address it in the question session. What the webinar is about is how we developed a system where we think we can greatly increase the throughput of calcium contractility research. I think that is going to be a great help in standardizing calcium contractility experiments, which I think everybody who is involved in these experiments understands the importance of. Myocytes, isolated myocytes have been around for a long time. When I did a little bit of research, the oldest publication that I could find was Powell 76 that showed how to isolate calcium-tolerant myocytes. Later in the 70s and the early 80s, it got picked up by a few notable labs, the Thomas Smith, Ralph Keller lab in Boston being one of them, the Isenberg lab in Germany, and I'm sure I'm forgetting other important names, so please excuse me for that. The technique caught on. Because there was demand, some companies started to make equipment for it. The first box that allowed you to do video-based edge detection and to measure contractility at a high rate was from Crescent Electronics. People who have it or have an old Ion Optics system will probably recognize this bit, where you can switch the frame rate between 60, 120, and 240 frames per second. That's the same camera system Ion Optics used till about 10 years ago. And at least as important, in 1985 was the publication of the seminal paper from the Roger Chen group, where they described new cell-permeable calcium-sensitive dyes. And this finally allowed you in a reasonably straightforward way to measure both contractility and calcium. With the advent of faster computers and Microsoft Windows, instead of an analog edge detection system, it got switched to a video-based edge detection system, which you can see here. So you have a direct confirmation whether you get good calcium transients and good cell shortening measurements. And later in the 90s, this technique had been developed separately in different laboratories on the world. The Garnier lab in 93 had a publication on it, Grenzier and Irving in 95. That's the lab where I worked. And Ion Optics picked it up and made a real-time system for it. It was too late to help me with my PhD, but it did get me a job at Ion Optics. And by the 2000s, the technique was well established and more labs started to use it. The pros became apparent. It was a very good assay to study drugs, disease, or mutations. I mean, the 2000s was the era where transgenic mouse models really came of age, and it was used a lot for that, and to study the effect on excitation-contraction coupling. The downsides also became clear. It's really important to have a good cell isolation. Your results are only as good as the cell isolation. And it also depends what kind of cell isolation you do. During the isolation process, a lot of dephosphorylation of proteins takes place, so the cells are not exactly the same after isolation as before the isolation. But what exactly happens Largely depends on your isolation protocol. Over the past years, I think there has been a conversion of isolation protocols, so the situation is better than it used to be. But still, I would rather have that there were just a few well-established protocols that everyone is stuck with. What you can do is, of course, you can look at the results from your data, and that you can compare with other labs. For example, resting sarcomere lengths are a very good indicator whether your cell isolation is good. But anyway, the next thing is cell numbers. Not being able to collect a lot of myocyte data from a lot of myocytes has always been a problem. You're collecting myocytes from the whole heart, or at least from the left ventricle. There's just a lot of variation that you can't always account for if you don't do enough measurements. The next slide is about a study that happened just before I joined the laboratory of Carl Epstein and Rongli Liao. And it shows you both the beauty of the system. This is a standard isoproterenal effect to study the effect of beta-adrenergic stimuli on hypertensive rats. And you can see a very nice dose response curve, which is blunted in the animal model, in the hypertensive rats. What it also shows is that you need a really big difference before you can establish a significant difference. And I was in the lab at the time, and I know getting these graphs actually took months to get the variation down as much as possible, to control the experiments fully, and to get this data out of it. And at the time already, when I was sitting in the dark for 5 hours doing this kind of experiments, I was already thinking about ways to automate it. It has never happened so far. And why? Because the requirements of the experiment don't really lend itself very well to making a high throughput system. You can go to a 5 times objective and then you see 100 cells, but you don't have the resolution to measure calcium well, or to measure contractility with high resolution. You can probably see it contract, but it's like a binary response. They contract or they don't, but you are interested in the kinetics. And I am definitely interested in getting to see sarcomere length. You don't see sarcomere length, you don't have the resolution until you go to an objective with a numerical aperture of at least 0.75, and it limits you to 20 or 40 times objectives. You also need high temporal resolution. If you're below 200 frames per second, you're going to miss the peak of the contractions of mouse or rat myocytes if you're at 37 degrees. For calcium, you need to go even higher. So calcium imaging is actually difficult. And the last problem is every cell has to be followed for a number of seconds. So you have enough transients to know whether it's a regularly beating cell, or in case you're interested in arrhythmias, whether there's an arrhythmic event. So all these things conspire against each other. But considering you have these design constraints, how far could you get? Suppose you measure each cell for 5 seconds and you pace at 2 Hertz, it gives you 10 contractions, enough to find out whether it's a normal cell. Say it would only take you 3 seconds to find the next cell. With the current system, you have to switch between the microscope and the screen, you have to rotate, you have to adjust the diaphragm. It takes between 2 to 5 minutes to switch between cells. So if you have 20 minutes per well, 1200 seconds, you would only be able to do about 10 cells. But if you only need 3 seconds to find the next cell, then you can do 150 cells per well instead of 10. And if you would be able to keep that up for 4 hours, 4 hours of measurements, 12 times 150 would be 1800 cells a day. Now that would be serious progress. So yeah, theoretically it's possible. The question is, can we do it? So for a high throughput system with serial measurements, we need speed to minimize the time between measurements. So you need fast hardware, rapid movement of the optics or the cells. You need high-speed software. Luckily, Ion Optics already has that, so we could lean on that. And then, of course, you need to improve the analysis, because if you stick with the currently available analysis methods, you're just moving the bottleneck. So I'll address each of these 3 things. It's a big project altogether. And after discussing it between the different groups, the VU Medical Center, where I hold a part-time position, was very enthusiastic about it. Ion Optics was enthusiastic about it. But we decided it was good to start fresh, so we could put in new ideas and not be constrained too much by the past. So we set up a separate company, Cytocyber BV. It's in Amsterdam. I get space from the university to do the work. We have a nice team together now. We decided to put our thoughts into practice. How do you go fast? Moving a dish with cells fast is not really a good option. the solution will start to slosh, the cells will start to move. So you actually need to move the microscope. Now the microscope is a bit heavy to move fast, but all microscopes these days have infinity corrected objectives. What it means in principle, the distance from the objective to the first lens doesn't really matter. So what you could do, you could make an X, Y, and Z stage where the objective moves instead of the cells, and then just measure it on the camera. You just have to design the right optics. So we first did an optical bench experiment where you put a camera together with some lenses. Here you have an objective that can move back and forth, and this is 2 pictures taken at different distances from the camera. And indeed, the magnification does not change when you move the objectives. Yes, the microscope companies were right. It is properly infinity corrected. Then we decided to see if we can actually build a stage that allowed us to grab in movements, and it was the first proof of concept, if you will. And we could move to the next field of view in about 30 milliseconds with 50 nanometer precision. So we thought we had a pretty good concept to build a microscope from. So we went to the next phase and we actually built it. It's a box. The nice thing about a box is that you can make it light tight and you can control the temperature. But it has a universal coupling. This here is a C-mount adapter, so you can connect anything to it, from a camera to a confocal system. There's an epifluorescence port on the back, and of course, the inevitable control box. If you open it up, you see we can put a 35 millimeter dish in, what we do here, but you also have the space to put in 35 millimeter dishes. It's nicely temperature controlled. We're about to add humidity and CO2 control, so then you have almost an incubator where you can do the experiments. What I'm going to show you next is, I'm going to show you 4 movies that explain the operation of the microscope and what you can do with it. The first one is actually, you don't have oculars anymore, so you need to have a way to navigate through the microscope. We only have one objective, so we have to figure out how to give you a proper overview. These things are being shown in the movie. So here you find the region that is where the cell will be, and then we can click around this window to go to different cells. We can click around in the overview if we want to a different part of the dish. This is the overview of the whole field of view that we have. So this is about the area where you can see cells in the 35 millimeter dish. We can click in this dish and we'll retile this image in a new position. So you click on it and you see how rapidly it moves to different positions. You have a good overview, you can see where the cells are, which is not a problem in this case because we have plenty of cells. But if you have sparsely populated cells, you have to find them. For the way to get overview, so you can find the cells that there are. Let's go to the next movie. Manual cell finding. We decided to first define a way where you could manually find the cells and see how much improvement that gave us. There are always going to be situations where you want to manually find the cells. So you click on the cell that you're interested in, you rotate the cell using your mouse or your keyboard. If you find a cell you like, you add it. So now we find one cell, and you see this red rectangle. That's the cell we have already found, and it stores the position, it saves the image, so you always have a backup image of the cell that you've been studying. Then you go to the next cell. The dexterity it requires is a little bit like a computer game, so I find that interns are actually better at this than people who have been using the system for a long time, just because they're faster. We'll solve that when we actually automate the full system. We already have made a start on it, and we think actually in 2-3 months' time we can do the experiments where you walk away from the microscope and you come back, and hopefully it has collected many good results. As you saw in the manual mode, you're doing the same thing again and again. You click on a cell, you judge whether it's good, you may adjust the autofocus, then you rotate it a little bit and you check whether the signal is good. All these steps can be automated. So that's what you will see here. It first checks in the full field of view, where you're now. It sees which of the cells are actually beating. Once it has found the beating cells, it'll put them one by one in the window here. It does an auto-focusing algorithm to find the best plane where you get a good sarcomere structure, and then it adds it to the cells here. So you can click through it and you can revisit the cells you had already found. I'll do it one more time. So here it's finding the cells that are actually beating, and then one by one it goes through the cells it's found and readjust where necessary angle, focus, and position. Once you've done that, you can actually go to the data collection. The first part of the video, we adjust your sarcomere length. So it's actually a dual monitor system. On the right side, you see the window that you navigate the microscope with. On the left side are the actual measurements, which is the good old ion optics sarcomere length program. And see, we can rapidly cycle through the different cells. Each one is measured for 5 seconds. And then you click on the next cell and you measure that one. We're busy in the process of fully automating this part. And then it will literally be a fraction of a second before you're at the next cell. Same thing you can do by also collecting calcium. These weren't great cells, so my apologies that the tranches are not as nice as they could be, but it shows the principle. Here again, you see the manual selection of the cells. And this is a real-time workflow where you find a cell, you adjust it, you add it to your list, and then you start a measurement. So that's what we can do. We made a user-friendly interface, so it's easy to get started with the system. What we do is we actually take interns and we give them a very summary overview of how the system works, and then we just let them go. And that seems to work quite well. It's given the impression that the system is relatively straightforward to use. So you can select the cells manually. You can use the mouse or you can use the keyboard shortcuts, just like you would do for a video game. We also have an algorithm to automate the cell finding. And you can measure each cell once if you want to measure as many cells as possible, or you can do repeated measurements on the cells. What we have not lost is the high definition measurements of calcium and contractility. To put it in perspective, these are the data from an experiment we did last week. We decided to time it just for the sake of it. The part that still takes the longest time is the cell isolation, preparing the solutions, getting the animal. These, by the way, this would be red data. Then the cells need to be plated. I like to let them adhere to a laminin and let them stabilize. So this particular day, we didn't actually get to doing measurements till 3 o'clock in the afternoon. Minimum 24, fast hands. He managed to collect contractility from 182 cells in 45 minutes. At 4 o'clock, he handed on the system to the next user. And that's the beauty. If you don't need that many cells, it's very easy to do multiple projects in parallel because you will always have enough cells. And it doesn't take that long anymore to collect the data. So we cultured the cells overnight, and the next day we measured them again between 10 and 12. This time it was Diederik, 35 years old, a little bit slower than Menon, but he still managed to collect 227 cells in about 2 hours. Then he analyzed the data from day 1 and day 2, and that took him exactly 2 minutes. He took another 15 minutes to make the graphs, and at 12:30 he was ready to hand the system again over to the next user. I'll go back to the analysis and how we actually do it. So these are the results from these 2 days. What I actually wanted to do is I wanted to check what the difference was in cell behavior between day 1 and day 2. On the top left, you see the data from day 1. It's a nice normal distribution. It's a frequency distribution. It looks like on day 2 that the contractility has slightly increased. The distribution has narrowed somewhat. So you could argue it's a nice and stable preparation to do experiments on the next day. Here we overlap the two, so you can indeed see that there's a bit of a shift to increase contractility. And what I was personally curious about, and I never really looked at it, I wanted to know is there actually a correlation between your end-diastolic sarcomere length and percentage shortening? Well, the answer is no, which is actually nice. It's nice and all to collect, what was it, 400 cells in about 3 hours. But you need to analyze them. And people that use existing synoptic systems, that even a file like this, where you have 1, 2, 3, 4, 5, 6, 7, 8, 9 cells, that would easily take you 15, 20 minutes. Plus, you have to check your data carefully because it's not always that secure in baseline detection, and there could be trouble with the exponential fit of the return of the calcium trends, etc., etc., etc. And you have to do the files one by one. Then you have to paste them in Excel and so forth. So what we decided to make a program where you could load batches of cells. There would be automatic rejection criteria. If you have artifacts, we just throw away the data for you. We improved some of the algorithms so we always have a stable baseline, and made some other improvements as well. And then with one click, you can export the data to Excel. This is just a screenshot of the program. We first made a standalone version of the program that can run on your PC, and then we decided to make a cloud-based version that I'm really going to push on you later on, but I'll get back to the discussion of that. What I will show you is actually a movie of how we do the analysis, and in case you're interested, I think we're about ready for beta testers, so send us an email if you're interested. So the data analysis. You have to sign in, of course. This is the web version. You have to upload files. You select the files you want to measure, in this case 3. You open it. You have to add actually a few constants. Upload it. To upload it actually takes some time depending on how many files you load. We saved that time by cutting it out of the movie. In the case of the 400 cells, that is the part that took slightly less than 2 minutes. If you have less cells, then it takes 10, 20, 30 seconds. And of course, that also depends on your computer and your internet connection. Then we open one of the files. Look, for example, at Myocyte 1. Downloads graphs. And here you can see the contractions that are in red, they're actually rejected. And as in this case, of course, there's actually artifacts on the trace. And here, probably because we moved away from the cell before we were ready, you see a lot of noise and that part gets rejected as well. You can zoom in on the single transients, and here you see your, oh, not so fast. So you can see your fit of the calcium transient and your fit of the contractility transient. And then when you're done, you click on download. You can download the Excel file or the CSV files. I think it asks you some additional questions, whether you want decimal points or commas, depends on which country you live. And when that's done, you open it in Excel, and there's your data. In this case, it's just average data. You can also export the transient from every single, export the data from every single transient. And then here are your trusted parameters like the departure velocity, the percentage shortening, time to peak, and we have time to return. The parameters that you're used to, and a few extra. It has actually been properly validated. It gives you the same results as you're used to, well, except where, of course, we improved it. Actually, we added a new feature to it that didn't exist before. It turns out everybody that uses the system wants to know whether the cells are arrhythmic. Some people want to exclude this data. Some people want to score how many cells are arrhythmic. And especially pharmaceutical companies are very interested in that part. For contractility, it's not that difficult to detect clear arrhythmias. We can already do that with our regular program. It turns out to be difficult when you have a noisy calcium transient with a noisy blip on it. It's very hard to catch the rules. It turns out in this age of deep learning, it's very easy to do with machine learning. So we made a training program where if you want to catch a particular type of odd transient, and of course often enough, You give us a number of transients where that oddity occurs. We make the classifiers, you put it in the program, and off you go. It will recognize all the strange irregularities that you can think of. It's actually pretty cool. That's why I wanted to show it to you. So let's get back to the slide that I showed you before, where you have an ISO dose response curve, and see how nicely you can measure it, but you have a problem with the amount of data you can do. This is what we can do now. So I'm only zooming in on a small part of the curve, to very low concentrations. And these are the results from 1 day. A lot of control cells, then a lot of cells with 2.5 nanomolar going up to 50 nanomolar. It's interesting, even if it's just 1 day, you see there's an immediate effect. And actually, fractional shorting is not the best way to measure the effect of ISO. The kinetic parameters are affected much more quickly, but I wanted to be able to compare it with the old graph. It's also interesting what you see is when you move up, where here there's just a lot of variation. It's probably nicely normally distributed. If you start to move up, you actually have responders and non-responders, which is not that shocking since the cells are from everywhere in the heart. But it is very complicating if you don't collect that many cells, because then you can't show that you actually have a bimodal distribution, and you have a lot of variation that you can't really account for. So I think it's very interesting to relook at a lot of the results in the literature, but then with a very high number of cells, and see whether you actually have different groups of cells in the heart. Another thing that you can do is do repeated measures. This was a simple experiment. We just followed 10 cells. We added a creatine kinase inhibitor. It's supposed to kill the cells. We know that from past experiments, and it nicely does. You see the cells slowly deteriorate, where they contract less and less and less. Diastolic sarcomere length goes down and down, indicating you start to get calcium overload. Then about half an hour, they all die. Except one that just keeps beating and beating and beating. I find it very intriguing, because now, of course, what I want to do is find a way to pick up the single cell and see why it is different from all the other cells. So that's more or less where we stand at the moment. We can do, without pushing it too much, we can do 800 cells a day now. In automatic mode, when you don't have any user intervention, we now are at the point where between all the accepted and rejected cells, we average 6 seconds between cells. It's not exactly the 3 seconds that I showed you in the beginning, but it's a good start, and I think there's room for improvement. And of those cells that are found automatically, 80% of them actually already give good measurements that, yeah, you can just take for your analysis. We're still not compromising on the calcium and conductility data. The temporal and spatial resolution is the same it has always been. I think we have achieved a more than 95% reduction in the time needed for data analysis. And what I think is really cool is that there's new ways to look at the data. I always wanted to look at the distribution of the data in the population, not necessarily just at a bar graph and hope that it is significant or not. And I think we have the chance to do that now. To get back to isolation quality, if you can make a distribution diagram, you can make a decision whether the population is the same as it was the week before. So you can make an informed decision whether to accept or reject the data from that experimental day. Or if everybody does it, you can compare between different labs, or you can compare different methods. What's next? As soon as you've solved the one bottleneck, the next bottleneck appears. What we're working very hard on now, and we're just delivering the first systems, is actually ways to do multiple dishes. If you have just a 35 millimeter dish and you have to take it out, you put the next one in, and you have to wait to let the cells settle down again. It will be much easier if you can just go from well to well, especially if you want to test different compounds. So we made an automated perfusion system where we can address each well. We have a pacing system where you can address each well individually, so that you don't have to open up your microscope for several hours if you once you get going properly. There's work to be done, but it's already working, and it's good fun. Where do we want to go from here? I think it's going to be very interesting to increase content. If you can get a phenotype, contractility, we'll be very interested to couple the phenotype to what is actually happening inside the cell. Calcium is just one example, but there's no reason why you couldn't do FRET experiments. What I find an interesting thought is you measure the cells, you know the exact location, So you could do immunostaining, antibody staining, and go back to the same cells and look what is actually happening to your protein of interest. One thing would be very cool is to be able to pick up the cells and then do single cell sequencing, single cell proteomics. Of course, instead of calcium, you could measure photosensitive dyes. There's some very exciting dyes coming out that should make it much easier to actually do good optical action potential measurements. Of course, I want to further increase throughput. I think we can easily double it every year for the next few years. We already have ideas how to parallelize the experiments. So I don't think we're at the end yet with 800 cells per day. And I think the really cool thing would actually be to start a cloud platform. That's why I put it in the cloud, and that's why I want to strongly incentivize people to actually use it. Because I think it opens the road to create a cardiac myocyte data repository. I don't know how the situation is in the US or other countries, but in the Netherlands, starting in 2020, it's mandatory for universities to make the data public that have been published. So every university is now making their own database. But that makes it hard to search for. So what if there would be a database where one click of the button, you can make your published data public, and other people can look at it, and you have the tools to look at it carefully. For one thing, you can compare quality of cells. It makes the data accessible. Modelers that do in silica work would love it, because they're always looking for more cells that have gotten a certain treatment. Arguably, it's also much better use of animals. What if you want to do an experiment and you search the database and you see the experiment has already been done, and you look whether it indeed has what you already want to know? Once enough properly annotated data are online, well, there's going to be some clever person that's going to figure out how to extract more information from it. Like, if you see this and this and that with that and that and that, can you target the gene involved? I don't know, but someone will find it out. And that's why I'd like to start this. Anyway, wherever it goes, I think we have exciting times ahead of us. And that's about the end of my talk, except, of course, I'd have to acknowledge the people that work with me. First of all, the VU Medical Center, where Diederik Custer has been very supportive in giving me access to cells, and Professor van der Velden, head of the department, that gave her full support to the project and letting me work on it. Max, without whom we wouldn't have the nice cells that you saw in this presentation. Cord and Ruth, interns that did most of the data collection. And from Inoptics, I couldn't have done it without excellent software support from Tom Udale, Leo, and Sander.
So first, Michael, for you, in your opinion, how you brought this up? There was a lot of discussion about myocyte isolation through the registration process. So the question is, how can we isolate better myocytes and make better use of them? Can you share some tips, tricks, and suggestions for us?
That's a wide-ranging question. I think a lot of an improvement has been made over the past years with good protocols that are also relatively straightforward for others to follow. I mean, the advent of liberase has greatly improved things, but there are also very good collagenase type 2 protocols around. Cell culture has also been improved. I mean, we see that on the second day after isolation, the contractility and the appearance of the myocytes is about as good as on day one. We used the protocol from Peter Lip in Hamburg. I think we used cytoscalasint as an actomyosin inhibitor to prevent the myocytes from hypercontracting, and that made a major improvement. I know there's also other protocols out there. We recently found a protocol that actually doesn't even use a Langdorf perfusion, which would again make life easier. But there are many ways out there. Circumstances differ. What there actually should be is there should be some assay to test certain aspects of the cells resulting from the isolation to know that you're on the right track or not. That's maybe at least as important. I'm not sure if Joe has to add something to that.
No, not really. I mean, there's always a lot of variability. At some point, there needs to be somebody that does kind of an overview of different isolation techniques and actually publishes something that addresses, how you would get the best myocytes, but it's hard to find that information. Every lab seems to have varying results.
To add to that, even with our system, we find that the higher source of variation is still inter-experimental variability between different isolations. So it's an ongoing struggle to get it right.
Mm-hmm. I think your
This
point.
also,
go ahead, Joe.
Go ahead, Eddie. All right. I was going to say this also points to doing higher throughput, so that variability kind of goes out in the wash, and you can get more information, even with variability from myocytes. The more information you collect, the more you can make use of those cells.
Mm-hmm. No, and your point, Michael, about finding tests to standardize or evaluate the isolation methods, downstream tests, are equally important, right? To determine what is working.
What a high throughput system allows you to do for sure is to do a baseline study every day you do an isolation. So it took us 45 minutes, and I think you can improve on that. And then you get a frequency distribution to see whether you are in the ballpark with your other isolations or what other labs publish. That could become a standard curve.
Very good. Great thoughts. Perhaps after the webinar, we'll have to think about some resources or things that we might be able to direct the audience and those that registered to on this subject, because it sounds like from the way you guys have discussed this with me in the background as well, it's an ongoing and important discussion to have as part of this research. So next question. What are some of the ways that we can address the variability we see in both data acquisition and analysis? So again, Michael, you spoke a bit about this, but maybe you can cycle back.
There's a couple of sources of variation after you take out the cell isolation itself, and that is one important one is environmental control. One of the weaknesses of the existing iNoptic system is that you have to use perfusion to keep the temperature constant, and it's hard to keep it really constant. And if you don't pay attention, your temperature will differ, which will greatly affect the kinetics of your data. So being in a box where you can guarantee a constant temperature will greatly help, especially if at some point we add CO2 control and humidity control. Then you can just leave all your cells in the microscope for a long time. You don't have to disturb them anymore. And I think that will actually extend the duration of your experiments, that you can do measurements. The other thing is that you take out the user bias by automating the experiments. That has an upside and a downside. If you take out the user bias, you also take cells that you maybe otherwise wouldn't check, like that are very contractile or have very little contractility, but at least you take out the bias. And if you do enough cells, they just become the tails of your distribution. So that's all right, I guess. And the last thing is, there's a reason why medicine studies are done double-blind, because no matter how hard you try, if you know what you want the outcome to be, you're biased. But if you fully automate the analysis, there won't be a bias anymore because you can't cherry pick your data.
Right.
So I think that is not a contribution where it just does it and you just accept the rejection criteria that are set.
Those good points. Joe, do you have anything to add on that one?
Just reiterate the what Michael mentioned about temperature control. There is likely be inherent variability in the myocytes, and there's nothing that we could or should do about it. It's just the way they are. But everything beyond that is some failure in the system, the way it's set up. By controlling temperature better, we're eliminating one of the variabilities that we see in our low throughput systems, and that is small fluctuations in temperature have an effect on the contractile performance of the myocytes.
Okay, very good. Next question, this one's from Frederic, and the question is, in your experience, how reliable are measurements of cells with irregular sarcomere structure? And an example here would be iPSC cells, or cardiomyocytes, I should say. Can you comment, maybe Michael?
You have to accept that they are somewhat immature. They are immature by definition because they don't have a very regular sarcomere structure.
Right.
That being a given, the amount of organization is determines how regularly and how well they contract. And if you look at the cell while you're doing a treatment, you still get an assessment of whether you improve or make contraction worse. Don't forget, we're not looking for absolute numbers in most cases. We're looking at an assay where we see whether something is changing or not.
Right.
In adult myocytes, it's easy because you have this very regular pattern, and you have some absolute data to go with. But even in the neonatal myocytes or stem cells, by looking at changes when you add a drug, you will be able to find whether there's an effect, and kinetics as well. So yeah, you can use it even if you can't measure sarcomere length. You just have to adjust your thoughts and realize it's an assay and it's not an absolute measurement.
Right. Right. Okay. So basically just larger or wider confidence intervals in that type of application, and just has to be worked into the scope of the project.
Yes.
Okay. Very good.
We're fully planning to adapt the protocol to neonatals and stem cells, but it's not a first priority. Our first priority is the area where it's needed hardest, and that is the adult isolated myocytes.
Okay. Very good. Next question, Stephen Marston has asked, is it possible to make successive measurements on the same set of cells after adding different doses or drugs, for instance, an agonist?
Absolutely.
Okay.
It's critical that you control your environment very well so the cells survive for a long time. And one of the key things is to let the cells stabilize before you measure them. We stabilize them by plating them on laminin and let them sit for an hour. And then we put in the microscope, and since they don't move, I'm actually used to, from my experience, you can do with thyroid, you can do 20 minutes experiments max before the cells start to deteriorate. But it turns out not to be true. If you don't move them and the temperature is very constant, we actually did dose response curve with the same dish for 4 concentrations of isoproterenol. That's not the data that I showed you, by the way. And the cells were fine too. They kept beating and beating for an hour and a half, 2 hours, and we couldn't find a noticeable difference. So I think you can get much more out of your cells than you could in the past just by controlling the situation better. I think actually moving the dish is quite detrimental to the cell. So that having said, it is entirely possible to measure a well, incubate with a drug. In the meanwhile, you can measure the next well, and when you've measured the next well, you can return to the first well and measure the effect of the compound.
Okay.
Just to piggyback on that, Michael, one of the things that we're working on now is to introduce CO2 so that we could move away from thyroids and use bicarbonate buffered systems. The cells seem to do much better in bicarbonate buffered systems as well.
Perfect. Very good. And actually, this question relates. I think it does. Something that's come in from Frederick again. How does pacing and perfusion in the system work at the moment?
Depends whether you use one well or a multi-well plate. With a one well, we just have an inlet and an outlet for solution. You're in a microscope, so you can't see what is happening. So I didn't want to have a perfusion system where you use vacuum to siphon off the solution, because everybody who does the experiments knows occasionally that goes wrong. And I don't want something to go wrong that I can't see happening. So instead what we move to is to have precise pumps, and we do balanced flow. So we know exactly how much we put in, we know exactly how much we take out. And by having multiple valves on the pump, we can switch solutions or switch between wells.
Okay.
The pacing, we just literally put a box in between that allows to select which well is being paced. So this is a non-issue if you just have a single dish. But if you have a multi-well plate, we can address each well individually. Right now up to 8 wells, but we'll improve that in the future.
Okay.
It's also worth noting the microscope itself has a sensor in it and a drip tray in case something goes sideways. So even though you can't see it, there are some fail-safes there to prevent damage to the microscope.
Okay.
But still, I'd rather not spill.
Yeah. Very good. And actually, I'm on this note as well. Is it the case that the pacer is included in the microscope setup, or is that a separate part that needs to be added onto the system?
It's a separate part.
Okay.
We haven't been thinking about it, and I'm not excluding that it will happen in the future, because it's actually not all that difficult. But we started the company in September 2015. We have just shipped our first system. So what we did is we made the absolutely minimal viable product that gave us really good results, and we decided to worry about the peripherals after that,
Mm-hmm.
which we're addressing right now.
Mm-hmm. Okay.
But the system does include a field stimulator.
Yes, correct.
It's just not built into the microscope.
Correct.
Right. So effectively what you're doing now is you would be compiling a system with the necessary components, but you see an option to incorporate some of these in a future version of the product.
Yeah, it makes sense. The less boxes clutter your desk, the better it is, I think.
Right. Okay, a question from Lily. Just, is it the case that only a 35 millimeter dish can be used in the system?
No, so far we have used two types of dishes.
Okay.
We have used one particular 8-well dish from IBD that works well because we can, without moving anything, we can actually observe 4 wells at the same time.
Okay.
And we're working right now to automate it for a 24-well plate, but we're already doing measurements with the 24-well plates.
Okay. Do you see any expansion in this area just to accommodate different types of plates, or?
Yeah, in the end, it's once you have a motion system that can move large plates around, you can use any dish type there is. So I wanted to make as generic a system as possible, so that you definitely shouldn't be stuck with buying consumables from us.
Right.
Because I don't think that's very nice, because other people are much better at making consumables. So I wanted to get a situation where you can use what you're used to using and what you think is perfect for your experiments, and we'll try to accommodate that.
Okay. Very good. Okay, another question. Well, this one comes from Mayal. And the question is, does the reliability of data depend on the force of the contraction? And if so, is there a threshold for this from your experience?
Hard question to answer, because of course, if you measure segment shortening, it's always a proxy of the actual force that is being developed. It doesn't necessarily translate. I mean, it's largely determined by the duration of your calcium transient. What I would say is that if you measure a lot of cells and you get your distribution, you always have the options in your further analysis to cut off the tails of the distribution and to stick with. That's kind of a statistical question, actually, that I would have to sort out properly. So far, it was kind of a non-issue because you didn't have the option.
Right.
So I'm actually, no, I don't have a perfect answer to that question, Mayel.
Okay, well, that would be a good one to include in the Q&A report. Give it some thought.
I will think about it and write it down.
Yeah, no, very good. How could these studies using the high throughput system be used? Like, in your opinion, maybe Michael, you could start. How do you envision this system supporting cardiac safety studies?
We have to make a serious effort in the ability to support measuring as many compounds as possible, so you can repeat the testing on multiple compounds. Let me rephrase it slightly. What you can't get away from with high throughput is that you still need to measure from a number of animals or a number of preparations. I'm hesitant to say animals. I know that the company in San Diego and Abayas is now doing it with human myocytes that they acquire on a very regular basis. So we're actually dealing with it this year because we got them a system to do this. So you still need to do it on a number of preparations, but you can, each day that you have preparation, you can test a large number of compounds and repeat the same number of compounds in the next day. So we try to facilitate as much as possible. So you can measure small batches, say 50 cells in a well, and then move on to the next well that or has a different concentration or a different compound. So you can look at cardiac toxicity effects. I mean, we're a long ways from being a true high throughput system where you can test thousands of compounds, but we can definitely move up in the chain, I think, of the way you test for it. But then if you want to test for cardiac toxicity, the animal model is at least as important as the actual measurements itself. We can detect arrhythmias, but the model has to be correct.
Right. Very good. Joe, any additional thoughts on that one?
Nope, Michael said it all.
Perfect. Okay. Looking just to get maybe a couple more questions in here. Can this be used on myocardial tissue to assess contractility on whole tissue or engineered strips? This question comes from Alan York.
Yes, there's no reason why not to. We didn't design it that way, but if you can move to different wells, you can also move to different wells that have engineered hard tissue in it.
Mm-hmm.
And even if we don't measure sarcomere length, we also have edge detection, so you can just follow the bending of the pillars. So there's no reason why you wouldn't be able to use it. And you can do fluorescence and contractility at the same time, whatever kind of fluorescence you're interested in.
Mm-hmm.
So I say the answer is unambiguously yes.
Perfect. And maybe our last question here. This comes from Alexia. Can we measure contractility on each cell of each of the cells for more than 5 seconds?
Yes. Of course, you just adjusted the protocol. I picked the 5 seconds for a reason, because we paced them at 2 hertz, and I think 10 contractions gives you a good image of the cell. But I know that people that are already using the system use, for example, 20 seconds, because that gives them the best chance of catching a rhythmic event.
Mm-hmm. So
you have to figure out what works, and of course, it's a trade-off between how long you look at the cell and how many cells you can do. But that's an experimental question. Yeah, you can set up the protocol any way you want. You can look at 100 seconds per cell. You just won't be able to do that many.
Sure. Okay, very good.
In that case, you're probably better off looking at 10 cells and coming back to the same cell every time and measuring for 5 or for 10 seconds again.
Okay. Very good. Any final thoughts, Joe, on that, or?
No, I think Michael was pretty clear. The experiment setup is similar to the existing Ion Optics software, Ion Wizard. You can define the duration of the experiments. There are new features that allow you to loop, so you can loop back to various epochs, if you're familiar with the software, mm-hmm. and that's simply just the duration of acquisition for each cell. So there's actually more functionality coming to make it even more expandable and more flexible.